Hello everybody and welcome to my new channel. So good news is that I remembered to turn on my mic very, very quickly. Okay, so what is this new channel? Let's try and get everything. Okay, so this new channel is sort of more of a way to make sure I'm doing my stuff, doing my research, doing my study, doing my reading of everything, as well as keeping you all up to date about my research project. So at the moment, we haven't quite yet got ethics approval for the particular project, so hopefully they'll pass it, but I can't see why they wouldn't. But ethics committee is a necessary evil. And hey, Math Pig, also everyone, thank you for coming. Hang on. Yeah, so I just want to say hello everyone for coming. By the way, Not Your Witness, Mama Atheist, Ben Millsabub, me, Dot Nerd, Jamie Mann, Gregory Whittaker, MoFab. I think that's everyone. If I've missed you, yell at me, by the way. Oh, Tria. Hello, Tria. Okay, so basically what this project is me, as everyone knows, I have 2 pet snakes and they're both adorable and lovely and I love them to bits. But my supervisor, she's doing microbial source tracking in wastewater and water catchment areas, and she said that what you do is you collect the water and see if there's any microbes in there that could cause illnesses such as E. coli and all of those really fun little bits. And Mama Atheist is a whiteboard. It says it's got nothing to do with it. It's just I'm at uni at the moment because I don't have much internet for this month. I've got 1 gig to last me 2 days, so I'm here. Anyway, so basically, what she said is, going with her work in microbial source tracking, she just had the idea of maybe we could do looking at E. coli variants in my snake's feces. So what I'm going to be doing is I'm going to be streaming once a week just to go over a paper that I read during the week and see if I can decipher it. The one that I'm doing today was written in 2008 and I can't remember the name of it off the top of my head, so we'll go into it very shortly. It gets a little bit boring and complicated in the middle, so I'll try and skip over that for you. But the beginning and the end are really, really interesting because microbes, E. coli especially, microbes in general, but E. coli has has sequences within its genetic code that is only specific to particular species. So, for example, the species, one type of E. coli variant lives in the gut of humans versus snakes versus birds versus rodents. So what they can do is actually trace some of those, using those genetic markers, they can actually figure out that, is it really choppy, is it? There's a big delay. Shouldn't be. I'm just going to, is it really laggy? Because if it is, I might actually restart it at a lower latency. I'm on ultra low latency, so I might actually just stop and restart again and go onto a different, if that makes sense. Anyway, so I'm just waiting for the chat. I'm just waiting for the chat to Okay, cool. Okay, cool. All right, no, okay. Sorry, I just realized that I left it on top chat rather than live chat on the because YouTube defaults to top chat, not live chat, because YouTube. Anyway, so basically, what I'm doing is I'm collecting the feces from my snake, and to see if the E. coli that I carry is being somehow introduced to my snakes, and if it is, then we can do other bits and pieces. So, anyway, that's basically the whole idea of this research project. And I'm really looking forward to showing everyone the research, do a little bit of science communication, talk about how freaking awesome microbes are, because they are. And not Jara's like, and I'm back. Did I miss much? Start again. I missed it. Don't worry. You just missed me going, oh my God, am I actually streaming? It's fine. Yeah. So I'm going to go and do try and read and decipher the paper, and hopefully once I get more into the swing of things, I might be able to get people who have been in the industry for a lot longer, like Scientist Mel or Dr. Stephen Hobbs, or someone coming in, or hopefully Lizzie Lang just coming in, and we can chat about it as well. So this is just my first solo stream, just to try and make it a little bit interesting, making life interesting. So I will go on to the next one. So this paper is no, that one. Okay, so this paper is called Assigning Escherichia coli, or E. coli, I can't pronounce that word, strains to phylogenetic groups, multi-locus sequence typing versus the PCR triplex method. So, yeah, that's pretty long and complicated. Darth Calculus goes, I've heard of endobind therapy exchanging gut bacteria. It's fascinating. It is pretty interesting. What actually got me into my university course was a person gave me a kombucha mother, and I was just like absolutely blown away about how weird this was, and then I started looking into the interactions of microbes with the gut, and it's fascinating. Gregory Whittaker, will you be only checking the snake's gut for the human E. coli? Yes, I'm not going to donate my stool sample for reasons that are reasonably obvious. First one is I don't really want to take some of my poo to uni. It's like, here's my poo, everyone. It's just not really thing. Will I see if E. coli will set up shop elsewhere in the snakes? I might actually have a chat to my my supervisor and see if I can bring in a snake shed and see what happens, because I do find that E. coli is within the gut of the snake from a human source, and if I do everything sterile and it's still not getting rid of the E. coli in the gut of the snake, then it's pretty much, we've seen what happens. Like, okay, cool, I'm giving them my E. coli and I don't know where it's coming from. I think there's also a good possibility that we could start looking at other other strains of microbes in there, and it's sort of a little bit pie in the sky at the moment. So you never know. We might be able to see if there's a change in flora just before the snake sheds or whatever. So hopefully by the end of the year, I might actually end up writing a paper and possibly, fingers crossed, publishing a paper on this. So, yeah. Nicholas Chop, kombucha, Japanese for I jizzed in your tea. Actually, I really should do a video on kombucha, and especially Gluconesidobacter kombucha, which is the bacteria that creates the pellicle, the cellulose pellicle on top of the kombucha, and sort of explain what it is, how it works, and yeah. Anyway, that's kind of going off topic. Do snake digestive tracts have an appendix? I don't think so, because I think the appendix started because of our ancestors consuming a lot of plant matter, and snakes throughout their evolutionary history have always had a carnivorous diet. So I don't think so, to be honest with you. All right. So basically, I'll try and break down the summary for you because it is complicated. I've read it a couple of times and I still don't understand a lot of it. But so basically, the summary says that it's recognized that E. coli consists of a number of distinct phylogroups and that strains of the different phylogroups vary in their ecological niches, life history characteristics, and a propensity to cause diseases. So in case you didn't know, E. coli actually lives pretty much everywhere. It's like Staph aureus. From what I understand, I could be wrong, but from what I understand, there's a golden staph is the common name of Staph aureus, which that kills people. It's not. It's the one that you can't kill with antibiotics. But MRSA is methicillin-resistant Staphylococcus aureus, and methicillin is one of our strongest antibiotics. So it's the same microbe, but it has that variant that stops being killed by bacteria by antibiotics. So Staph aureus does that. Okay, so E. coli is the same sort of thing. E. coli is fairly benign. In fact, we actually need it within our guts to help us digest our food, and it also lives on our skin. It lives literally everywhere. So when it gets into the wrong areas, such as your bladder, it can cause disease such as urinary tract infections. Most urinary tract infections start because of E. coli. E. coli is the reason why most people get UTIs. J.R. Eldridge, I opened the page and the first thing I saw was I jizzed in your tea. What a welcome. Okay, so going back to it. So a triplex PCR-based method that enables strains of E. coli to be assigned to a phylogroup using dichotomous key approach based on the presence or absence of two genes, CHUA and YJAA. So I'm going to call it Chua and Jizz. He said I needed to be professional, really. Anyway, and an anonymous DNA fragment, TSPE4.C2, so T-spec, I don't know, has been developed. However, the accuracy with which this method assigns strains to their correct phylogroup has not been adequately evaluated. So basically what they're trying to do is trying to make sure everything's reasonably within the same group and accurate groups. So consequently, 662 strains of E. coli were characterized using a multi-locus sequencing typing approach. So they just sequenced the E. coli strains, and then they try and stick them together to try and make a fairly reasonable phylogenetic tree. So the phylogenetic tree is the tree of life. So, you see those the famous pictures of like wolf and the branches down to dogs. So, and then as you go up, there's more and more branches. That's the phylogenetic tree. Gregory Whitaker, isn't there a variant of E. coli that was genetically modified to produce insulin? I think it was E. coli. I'm not sure, to be honest with you, but I do believe it was E. coli. So the analyses revealed that 80 to 90% of E. coli strains can be assigned to a phylogroup, and that 80 to 85% of the phylogroup members were assigned using the Clermont method are correct. So the Clermont method, I tried figuring out what it was, but I never quite got what it was. I couldn't find what it was, but I think the Clermont method was using different types of PCR methods, and just kind of like going, if this is positive, negative, it belongs in this group. So that it takes locuses of within the genetic thing. Is the E. coli my skin the same as my gut bacteria? They're the same species. I don't know about the same variant. Maybe, maybe not. Can you give me a moment, please? Can everyone please give me a few minutes? I'm getting kicked out because there's a class coming on. Hang on. How about now? Can everyone hear me? Can you hear me now? Hello? It's saying that you can hear me. Hello, hello, hello? Hello? Sounds like a toy phaser. Alright, hang on, give me a moment. True, it's not a stream until she forgets to turn on the mic. It was saying, I will say, OBS was telling me that you could hear me. But now I can't. Not Ja. Seriously. Okay. Alright, so what was I saying? That... Yes, we do need a Tech Talk Math Peak. Alright, so anyway, basically what I was saying, where was it going? Okay, so the Clermont typing actually figured out there was something like 8 different strains, I think. It said that 4 well-recognized phylogenetic groups that was designated A, B1, B2, and D. Basically, it means that they were able to use the genotyping to be able to designate those microbes into those particular figuring out those microbes and being able to designate those microbes into that particular grouping. So groups A and B1 are considered to be sister groups, and group B is considered by some to represent the ancestral lineage of E. coli. So B2 was a bit higher, is a bit higher in the phylogenetic tree than A, B1, and D. So the strains of the 4 groups differ in the phylogenetic characteristics, including their ability to exploit different sugars, the antibiotic resistant profiles, and the growth rate temperature relationships, which is a conversation that we can have a bit later. Anyway, so these different what's the word? Phenotypes. These different phenotypes, you can kind of sort them into, and they do it through genetic sequencing. And so genome size varies among the 4 phylo groups. Of course, that makes sense because if they've got different types of phenotypes, their base pair would change. The number of base pairs within the genotype would change. So your base pairs are UACG, and U is uracil, which is RNA. So your adenine, your guanine, your cytosine, your thymine, and then U for the RNA. The distribution or the presence or the absence of a variety of genes is thought to enable a strain to call extra-intestinal disease, and also varies among strains of the 4 phylo groups. So that's basically what they're saying. What I was saying earlier is that you can kind of track where they are and if they cause illness and things like that, going by the sequencing. So the strains of the 4 phylogenetic groups also appear to differ in their ecological niches, their life history characteristics and propensity to cause disease. For example, groups B2 and D strains are less frequently isolated from the environment, or fish, frogs and reptiles, than A or B1 strains. And in mammals, the B2 strains are more frequently isolated from hosts possessing hindgut modifications for microbial fermentation than other strains of phylo groups. So I'm not entirely... I think they're talking about B2 might seem to be coming from like ruminants, but from what I gathered, I could be wrong with that. And Finally, isolates recovered from extra-intestinal body sites more likely to be B2 or D strains than to be A or B1 strains. So I think it was, who was it actually said? I think it was, someone asked if, Gregory, is it, no, hang on, was it E. coli, Gregory or someone did ask if the E. coli living in your gut would be different to the E. coli living on your skin, and by the looks of this, it should be. So isolates recovered from extra-intestinal body sites more likely to be B2 or D strains than to be A or B1 strains. Thus, a great deal can be learnt concerning the characteristics of an unknown strain determining its phylogroup's membership. So it says here, Claremont and colleagues developed a multiplex PCR-based method that enables strains of E. coli to be, give me a moment, assigned a phylogroup using a dichotomist key approach based on the presence or absence of two genes. So that's your CHUA and YJAA, or Chua and Jizz, and an anonymous DNA fragments, So basically, if in this table here, if the Chua and Jizz was negative for that mutation, as well as T-spec, your phylogroup assignment was A, and that was J.I. Eldridge says depends on how well you wipe. That's true, actually. You probably, even though you would find the variant within your gut to be obviously more common in the gut, you'd probably still have that particular variant on your skin as well. Just because it's saying that it's not found commonly, it doesn't mean it's not found. So, yeah. And this is all part of your healthy microbiome anyway, so you don't have to worry too much. Talk to you soon, .nerd. So as you're seeing on the CHUA and all of that, that's how they are able to group these phylogenetic genes. And of course, because everything is complicated, you can have for the B2, you can have positive, positive, and positive, as well as positive, positive, negative, and still be in the same group. So what they're doing now is they're using a multi-locus sequencing typing for these particular isolates of E. coli, and they're trying to appropriately assign these particular E. coli groups, E. colis into those phylogroups using this particular method. So any questions at the moment? If anybody has any questions, let me know. I have a few questions myself. Is the sound okay? Because, hello Josiah, how you going? I just gotta get rid of that. Okay, that's better. Okay, so how's the sound? Is everything okay? All right, so basically what's happening here is they collected a whole heap of E. coli strains from various areas. So, for example, the first of these were collected from a diversity of host species in a variety of geographical locations, as well as from patients exhibiting different clinical syndromes. So other strains were collected in the Australian soil, sediment and water samples, so geography and that sort of thing. There's also German, I think strains from the Australian collection were characterised using the MLST scheme described at this particular website. And so they basically took E. coli samples from all over the place and ran it through these sequencing programs and stuff. Okay, So one of the problems that they had with the Claremont method is it only really looked at a small section of variation within the genetic sequencing. So the Claremont method had the potential to yield 8 distinct phylotypes from Schwa, Giz, and T-Spec. And it was and so the phylotype negative, positive was never observed among the 662 strains characterized in the study, and the phylogroup B12 phylotype positive, negative was rare. So what they did was they basically nucleotide sequence data consisted of this is where it kind of starts to get a little bit so much information I can't quite figure out what's going on. So according to the Claremont method, there was only eight types of variants. So what they did is that by using some more different types of methods, they were able to find more variants. So for example here, the Claremont method would correctly assigned 85% of the 91 strains to the correct phylogroup, which means that there was a 15% inaccuracy rate. So no strains with the Claremont B1, negative, positive, or B2 phylotype, all positive or positive, negatives were incorrectly assigned. But of the 22 strains with Claremont A phylotype of negative, positive, negatives, 9 was misclassified, and all of the strains within the phylotype negative could not be assigned a phylotype. Oh, don't know. There must be way more different types than that. I think there were different diets and cultures. Yeah. So basically, from reading this, they were able to make do a few more classifications. So because the Claremont type, even though it was well accepted back then to have those particular these 4 different types of strains, they're now actually finding that. They were finding more different types that didn't quite fit into those categories, so they started making new phylogenetic groupings. So here you've got A, B1, B2, and D, which were the original 4 from the Claremont. They've now added E, A, B1, and U, because they brought in another sequencing from what I understand of this paper. All right. So U denotes the strains that were not assigned to a phylogroup. So there's probably more different types of phylogroups within there. So as you can see here, the structure A, or BAPS, I'm not entirely sure what that meant. I again looked it up and I was like, I can't quite figure it out. So there's 20 strains within the structure of BAPS. B1 is 23, B2 is 23, D is 13, E is 3. Structure U in BAPS A was 7, U in B1 was 1, and structure B1 was BAPS 2 was 1. So as I said, U denotes a strain that was not assigned to a phylogroup. All right, so this is kind of when it starts to get a little bit pretty pictures. So as you can see here, this is the phylogenetic tree of the E. coli. So you've got the B1 here, and it shows how it's related to A and E here, D and B2. So they're all genetically related, but it's kind of showing the branches of the different types of E. coli going by the variants. So among strain relationships is found by near, what's it called? It was a near something. I can't remember, NJ tree, so it's by Notjar tree, based on 91 strains characterized using the French and German MLST schemes B2 strains are in red, D strains are in yellow, B1 strains are in green, E strains are in brown, and A strains are in blue. Strains not denoted by thin black lines could not be assigned to a phylo group. So as you can see, between B1 and A, there's a couple of thin lines, and there's one in E. So they're kind of like, can't quite figure out where it is. But but they were able to figure out where they've diverged, and they were able to make those things. So they didn't quite fit into a grouping, because nothing in biology it works. Not just that, yay, I have my own tree. It was near, can't remember, I keep thinking near journal. It's basically like a near junction or something. I can't remember what it was. Okay, so the groups recognized with the same 5 is recognized by structure, but the BAPS analysis subdivided group D strains, D1 and D2, and did the same for group B2 strains, table 4, which is here. That's table 4. So as you can see, it's starting to get quite complicated. Greater number. Okay. So they're talking about the structure, they're talking about that sort of thing, but here it is just showing that variance within variance can occur, which is understandable. It's evolution, like everything happens. Okay, so this is where it kind of starts. You have to use your brain and follow it, and it jumps around a lot. So me, being ADHD, had a lot of trouble following it, and at the same time, because of that, I'm not confident in explaining it to you, but from basically this is just the results. So what I'm going to do is, again, here we go, comparison of phylo group assignment of strains using the PCR-based Clermont method and the assignment of 225 strains using the nucleotide sequence data from the French MLST screen. So yeah, it's getting more and more complicated. The Clermont assignment has these positives and negatives. You've got the MLST assignment, and then you're starting to see over and over again, and it's getting more and more complicated. Among strain relationships is described by NJ tree based on the strains characterized by using the French MLST scheme. Here, again, just showing you that don't know if you need to worry about that many variants though. So again here, the MLST is showing, it's not actually saying which one's which, but MLST versus Clermont method. So there we go. That's again is kind of showing that the MLST is a lot more accurate, and gives more variance versus the Clermont method. All right, so I'm going to go to the next one. Bear with me. It's more or less saying the same stuff, and because once it gets to it's still saying the same stuff. But here is the phylogenetic tree. So here was the first common ancestor, and so that's first common ancestor of E. coli is about here. And as you continue going down, it starts showing you all of the different gene names. If you just keep going down, it'll show you. It's a knot-jar tree depicting the among strain relationships based on the 754 base pair portion of the CHUA gene. The label represents the strain name, then the Clermont phylotype of the strain. So CHUA, GIS, and T-spec, which is where 1 denotes the presence and 0 the absence of a PCR product. followed by the strains phylogroup group assignment based on the available MSLT data. Numbers at the nodes represent Bootstrap support areas. What's the logic behind the letters and numbers? Are B1, B2 more related to D and A? It did say right at the beginning, what page is that? That's 10. Nope. That's not page 10. It did say here that groups A and B1 are considered to be sister groups, and group B2 is considered by some to represent the ancestral line of the tree. No worries, Jamie. Have fun. I hope you're finding this interesting, because when I was reading it was pretty interesting, and then it got to the middle of it and I'm just going, ow. Anyway, so I don't know what the logic towards what they call the naming of things. Sometimes it just seems so arbitrary, and it makes life really difficult if you're trying to remember the name of a particular mutation. Next one. Okay, and that's... So I'm going to try... and go towards the end of... so just to wrap it up, because... this... and let's go to... Okay, so the foundation of the Clermont method. Okay. So the genes CHUA, YJAA, and the fragment TSPC4.C2 appear to have diverse origins. The GC content of a typical housekeeping gene in E. coli is 50.7%, while the GC content of CHUA is 50.6%, 52.4% for the putative lipase genes, and is 45.3 for... I'll just let me... YJAA. Thus, YJAA is likely to have been acquired from distantly related species. Although fragments similar to T-spec is present in Salmonella, CHUA and YJAA are absent from E. coli sister genus. The three genes are also absent from Escherichia ferganosi, data not shown, and the closest relative of E. coli, perhaps the closest Escherichia ferganosi, which is the closest relative of E. coli, perhaps indicating that the acquisition of the acquisition after the E. ferganosi E. coli divergence. However, as the phylogenies of these genes are congruent with most of the with the strain phylogeny inferred from the MLST data, the most parsimonious scenario is that these three genes were acquired early in E. coli's history and subsequently lost from some lineages. Of the three DNI fragments used in the Clermont method, CHUA appears to be the most stable. CHUA is present in all members of the B2 phylogroup and all but a single member of the D phylogroup. And this concurs with the fact that the gene is part of a large operon. The T-spec fragment is the next most stable, as it is present in a great majority of B1, B2 strains and is absent from the majority of D strains. The gene YJA is the least informative of the markers used by the Clermont method, as it can be absent from some B2 strains and significant fraction of the phylogroup A strains, and can be the only Clermont marker present in some phyllo B1 strains. Meaning that it's basically random. Well, not basically random, but it's kind of, you can't really rely too much on the YJA strain to be able to say this particular gene means that this particular E. coli strain belongs in this particular group. But I thought that was pretty interesting. They were able to use the genomes that they found when they sequenced it to be to be able to figure out the branching and where E. coli branched. And I think that's really one of the coolest things about evolution and genome sequencing, is that we're able to go and say, oh, this gene is found in the populations of this particular species, but it's not, but the sister or a similar species of the same genus would, that's where they diverge. That's when they can say, in this particular gene, this diverged and this one didn't. So a lot of creationists will point to, there's no such thing as a transitionary fossil. But the thing is that fossils are the weakest evidence that we have towards for evolution. It's our ability to sequence DNA is the reason why we are able to say this is when this particular species diverge, because you can see similar mutations within a particular species genome, that if that mutation is found in another species, but not in a different species, you'll know that, okay, so that species and that species diverged after diverging from this particular species. So I just think it's really, really cool that we can do this. So the concluding remarks is the results of the analysis presented in this paper suggest that improvements to the Clermont method may be difficult to achieve, as the gene content of E. coli is highly variable. J.R. Eldridge says, all fossils are transitional, everything is still evolving. That is true. There is no such thing as a transitional species or fossil. So the results of our analysis indicate that all strands exhibiting the phylotype of +, should be screened for the gene IBEA, as IBEA's presence indicates that it is highly likely the strain is a member of phylogroup B2. Among the isolates from Australia, 23% of strains exhibiting a ++ phylotype or IBEA positive. It may be difficult to identify a DNA fragment that is both unique to as well as common to all phylogroup E strains, as strains from this group seem to share alleles with many E. coli strains. For example, although phylogroup E strains exhibit distinct CHUA alleles, they share T-spec alleles with group 1 strains and GIS alleles from phylogroup A strains. Genome data on additional phylogroup E strains may allow the identification of markers unique to these strains. The results of this study demonstrate that for population-level studies, the Clermont method is an excellent technique for rapidly and inexpensively assigning strains of E. coli to phylogenetic groups. For samples taken from the feces of asymptomatic human hosts, so people who don't have an infection that's causing people to get sick, such as like a urinary tract infection or gastritis kind of infection that's caused by E. coli. So for samples taken from the feces of asymptomatic human hosts, the fraction of strains present in the sample that cannot be assigned to a phylogroup is likely to be low, and the fraction of strains that are misassigned to phylogroup is also likely to be low. Strains failing to yield any PCR products using the Clermont method should be scored as unassigned, and as strains with this profile are not monophyletic, they should be excluded from any statistical analyses. Further studies are needed to determine the validity of group A strains and E strains as phylogenetic groups. Sorry. Okay. I think .NET says, I think thanks to DNA research in some fossils, that's now how they've found some dinosaurs had feathers. Yep, I think that's probably likely. Don't know. I don't recall what methods were used to. You probably something like PCR. Maybe, I don't know. That's how it goes. Oh, well, thank you, Notja. Okay, so Further studies are needed to determine the validity of group A strains and E strains as phylogenetic groups as compared with phylogroups B1, B2, and D. Investigations to determine the boundaries of phylogroup D are also required. And I think... So basically they're saying that Clermont is reasonably accurate, but if we want to get a little bit more deeper in, there's other ways that we can look at it. Because PCR, while it is fantastic for sequencing and if you phylogenetic sequencing and stuff like that, MLST, for example, is a lot more accurate. So if you want to be a lot more accurate, then basically Clermont is what you start at, and then later continuing on with MLST because it's more accurate. Okay, so that was basically the long and short of this particular study. From what I can understand of that study, it was very, very confusing, very difficult to read because it's scientific paper. But that was basically it. That's how it works. And yeah, that's basically paper 1 of many, many papers to come of me streaming. So I hope you guys enjoyed that. Do you guys have any questions? Have you got any questions? Yeah. I just thought it was really interesting, personally, with that particular paper. I just thought it was really interesting that they were able to find 8 phylogenetic groups after using different techniques. But I also think from my research, I think they've now put it up to 14 groups, if I remember correctly. There's now quick question, did you guys find that boring? Do I need to make things more interesting? I'd love some feedback. I genuinely would. J.O. Eldridge, if you read the nucleoside sequence, you'll be speaking tongues again. Okay, well, let me have a go. Let's see if I can talk in my... Okay. FATCTGAATGGTATTGTGGCCTGGT RAGTT. Yeah, no, it does sound like I'm talking in tongues, doesn't it? Yeah, so if you guys let me know how you found it, if you enjoyed it. As I said, I will be continuing on releasing shorts whenever I can. I'll probably show shorts of me feeding my snakes. It might get a little bit repetitive. Every month or two I might say, do shorts again of something I've done earlier, because there's only so much you can do. But I'm really, really looking forward to this. I'm really going to be really interesting. I'm excited to get back into the labs. I'm excited to show you the results and see how we go. Bimils above is, I've understood somewhere in the ballpark of nothing that you've read or said so far, but I'm happy to give you the view and interaction. Well, thank you. I hope I was able to make it a little bit clearer. It is sometimes, I will say, it is very, very difficult sometimes to read something, at a university level and try to translate it into something that people who've done only high school biology is able to understand. And sometimes it's hard to do it because there's no way, there's no layperson word to explain it. So yeah, anyway, I thank you very much, everyone, for joining in, and I will let you go. And don't forget, scandium, iodine, nitrogen, and cerium.